Indeed, we presented yesterday at the poster discussion session the results of our sub-analysis of AURORA trial. AURORA is a molecular screening initiative led by the Breast International Group. And the aim of this initiative is to understanding more and more the heterogeneity of breast cancer. And basically, in this study, patients with metastatic breast cancers are enrolled, and these patients provide samples of their metastatic lesions and of primary tumor lesions, but also plasma samples, blood samples. And there is also, of course, a collection of full clinical information. And the idea is really to do this extensive profiling of breast tumors and to better understand how the disease evolved from the primary tumor to the metastatic lesion and across all the different treatment lines. So what we did in the project that we presented at ASCO was to focus specifically in patients treated with the CDK4/6 inhibitors. We know that for patients with hormone receptor-positive, HER2-negative metastatic breast cancer, CDK4/6 inhibitors plus endocrine therapy is the standard of care in first line. But we also know that not all patients derive the same benefit from this treatment. So the aim of our study was to identify clinical, pathological, and molecular characteristics that are associated with the prognosis in these patients. So overall, we analyzed 339 patients, and what we observed there are a number of characteristics associated with the survival in these patients. One first characteristic is very simple, it's clinical, and it's derives from the endocrine resistance sensitivity status according to the ESMO definition, the ABC guidelines definition, and we observed that patients with a primary endocrine resistance have a significantly shorter progression-free survival compared with patients who are endocrine sensitive, for instance, or endocrine therapy naive. And then from a molecular perspective, we observed that some mutations are associated with a shorter progression-free survival, and this is the case of a TP53 mutation and also acquired ESR1 mutation. On the other hand, for instance, a mutation of PIK3CA in our analysis was not associated with a different prognosis. And also in terms of histology, we observed that the ductal tumor and lobular tumor didn't different in terms of prognosis. So of course our data are very exploratory and should be considered as hypothesis generating, but I think they are very important in better understanding the heterogeneity in response to this treatment with the CDK4/6 inhibitors. And of course the idea is that if we are able to identify characteristics associated with a worse prognosis, this could help to design clinical trials to test also alternative treatment strategies. And I'm also particularly happy and proud to say that this project was awarded with a Merit Award from the ASCO Conquer Cancer Foundation. So I really want to thank all the people who collaborated in this project, from all the members of the CDK4/6 inhibitors working group in AURORA, and also the Breast International Group, who is the sponsor of AURORA.